Let us now look into the microscopic features of kidney. I am grateful to Professor Michael Horsch of University of Michigan Medical School for providing the images of histology sections used in this video. Kidneys are a pair of bean-shaped organs located on the posterior abdominal wall for excreting metabolic waste and maintaining fluid and electrolyte balance. Kidney is covered by a thin capsule made of collagen-rich connective tissue. It has an outer cortex and an inner medulla. Cortex is further separated into an outer zone and an inner juxtamedullary zone. In a coronal section of human kidney, renal medulla shows about 10 to 18 striated renal pyramids which are visible to the naked eye. Each renal pyramid has a peripheral base and an inner apex or the renal papilla which opens into a minor calyx. Renal cortex arches over these renal pyramids and the two are separated by arcuate arteries. Renal cortex extends between the renal pyramids as renal columns, and cortex is traversed by extensions of medulla which appear pale and they are called as medullary rays. Kidney is made up of uriniferous tubules and delicate connective tissue containing branches of renal vessels. Uriniferous tubule is made up of nephron and collecting duct. Each kidney has up to 1 million nephrons, and the function of nephrons is to produce urine. Each nephron consists of renal corpuscles that helps in ultrafiltration of plasma and various parts of the renal tubule that includes proximal convoluted tubule which helps in selective resorption of water, glucose and electrolytes, loop of Henle that helps in countercurrent exchange and multiplication, distal convoluted tubule and connecting tubule that help in active secretion. The collecting duct helps in concentrating the urine under the influence of antidiuretic hormone, and this concentrated urine is successively collected and passed through papillary duct, minor calyx, major calyx, finally to drain into the renal pelvis. Renal corpuscles, proximal convoluted tubule, distal convoluted tubule, connecting tubule and beginning of collecting duct are found in the renal cortex, whereas loop of Henle and most of the collecting duct and rest of the duct system is found in the renal medulla. Renal corpuscles are rounded structures found in the renal cortex. They have an outer Bowman's capsule and an inner glomerulus. This glomerulus is a collection of convoluted capillaries which are lined by fenestrated endothelium and a delicate mesangial matrix. Bowman's capsule is a cup-shaped blind-ending expansion of the renal tubule. It's invaginated by glomerulus. Its outer parietal layer is lined by simple squamous epithelium. Inner visceral layer covers the capillaries of the glomerulus and it is made up of specialized podocytes. Under electron microscope, these podocytes show primary food processes and secondary pedicles. Gap between these interdigitating pedicles of adjacent podocytes form the filtration slits. These filtration slits are covered by slit diaphragm through which the glomerular filtrate must pass to enter the urinary space, which is located between the two layers of Bowman's capsule. At the vascular pole of the renal corpuscle, an afferent arteriole feeds the capillaries of the glomerulus and an efferent arteriole collects the blood from these capillaries. At the opposite urinary pole of the corpuscle, the urinary space of Bowman's capsule continues with the lumen of proximal convoluted tubule. Glomerular basement membrane forms the filtration barrier. It is made up of fused basal lamina of epithelium and basal lamina of podocytes. Its interior is filled by delicate mesangial matrix with a few mesangial cells, and these mesangial cells have both phagocytic and contractile properties. Few extraglomerular mesangial cells or otherwise called as Lacis cells are also found at the vascular pole, and they form a part of juxtaglomerular apparatus. Bowman's capsule continues as the proximal convoluted tubule at its urinary pole. Proximal convoluted tubule is entirely found in renal cortex. It has a short straight neck at its beginning and continues as highly convoluted tube up to medulla, where it continues as loop of Henle. Proximal convoluted tubules are lined by strongly eosinophilic cuboidal cells with central euchromatic nucleus. Apical surface of these lining epithelial cells of proximal convoluted tubule show brush border as seen in this pus stained section, and their basal surfaces also show infoldings. Both these features suggest resorption function of the lining cells of PCT. Basal cytoplasm contains numerous mitochondria, suggesting active transportation against concentration gradient, and their lateral borders also show interdigitation with adjacent cells. Loop of Henle is a U-shaped segment seen in the renal medulla. It consists of thick descending segment, a thin segment, and a thick ascending segment successively. Thick descending and ascending segments are lined by cuboidal epithelium, whereas thin segment is lined by squamous epithelium. Presence of few cellular organelles in the cells of thin segment suggests passive transportation. Length of loop of Henle varies depending upon the position of the renal corpuscle in the cortex and whether the nephron is a short-looped or a long-looped nephron. Vasa recta form a network around the tubules of loops of Henle, and they help in countercurrent exchange of water and solutes. Loop of Henle continues as distal convoluted tubule in the renal cortex. Distal convoluted tubules are also lined by cuboidal epithelium similar to PCT, but they can be distinguished from the proximal convoluted tubule by having greater number of cells in the lumen. Their cells are less eosinophilic compared to the cells of the proximal convoluted tubule, and they have a distinct outline for their lumen, whereas the PCT outline of the lumen is fudged because of the presence of brush border. Juxtaglomerular apparatus helps in maintaining arterial blood pressure. It consists of three components: the modified smooth muscle cells of tunica media of afferent or less frequently efferent arterioles at the vascular pole of glomerulus. These modified smooth muscle cells are called as juxtaglomerular cells. The second component is the extraglomerular mesangial cells or the lacis cells. And the third component is the modified lining cells of the distal convoluted tubule known as macula densa. Macula densa cells are the osmoreceptors sensing the sodium chloride concentration. Lacis cells transmit this sensory signal. This activates the juxtaglomerular cells to secrete renin into bloodstream, which activates the renin-angiotensin-aldosterone system. Renin converts the circulating angiotensinogen into angiotensin 1, which is then subsequently converted into angiotensin 2 by the endothelial cells in the lung. Angiotensin 2 stimulates the zona glomerulosa cells of the suprarenal gland to secrete aldosterone, and this aldosterone in turn increases the sodium and water resorption from the collecting duct to increase the arterial blood pressure. Distal convoluted tubule continues as connecting tubule, which drains the urine into collecting duct. Connecting tubule and the cortical collecting duct are lined by squamous epithelium. However, the lining epithelium of the medullary part of collecting duct progressively increases in height, changing from cuboidal to columnar towards the renal papilla. They show two types of cells, the principal cells and few intercalated cells which are dark in color, and they are involved in hydrogen ion transportation. Urine from multiple collecting ducts is drained into papillary duct at the renal papilla. The papillary ducts subsequently open into minor calyx. 3 to 5 minor calyces join to form the major calyx. The major calyces open into renal pelvis. Minor and major calyces along with renal pelvis are lined by urothelium, and they show smooth muscle in their walls. Here we are seeing the major calyx showing the lining urothelium. So quickly recalling what we have seen so far, kidney has an outer cortex and an inner medulla covered by a capsule. Cortex contains renal corpuscles and cut sections of proximal and distal convoluted tubules, connecting tubules and beginning of collecting ducts. Renal medulla contains loop of Henle, terminal parts of collecting ducts which drain successively into papillary ducts, minor and major calyces, and finally into renal pelvis. Thank you. Hope you enjoyed this video. You can visit this site for similar histology videos.